Cloning is the process of making an exact copy of an organism. There are two types of cloning: reproductive cloning, which is used to make an actual cloned offspring, an actual baby, and therapeutic cloning, which is used just to collect stem cells. No baby is actually made. However, the process of reproductive cloning and therapeutic cloning is very, very similar, and it's both carried out by a process called somatic cell nuclear transfer. In somatic cell nuclear transfer, you need two cells. You need an egg cell from a donor. It can come from any female of that species. And then you need a somatic cell from the animal that you want to clone. So let's say you wanted to clone a sheep. You would need a sheep's egg cell, and then you would need maybe a skin cell from the sheep that you wanted to clone. You're going to empty out the egg cell. You're going to remove all the DNA in the whole nucleus so that you have an empty egg cell. And then you're going to take the nucleus from the skin cell of the animal you want to clone and remove it and put it inside of the egg cell. Now the egg cell thinks it's been fertilized, and it will act as if it has been fertilized, and it will create a zygote and then eventually a blastula, which is a small embryo. If you wanted to go through reproductive cloning, then you could insert this embryo into the uterus of a female of that species, and she would become pregnant. But if you just wanted to collect the stem cells, then you could take that embryo and collect the stem cells. And these would be pluripotent stem cells that have the potential to become many types of cells.